In this video, once again, we're going to talk about mismatch of pi orbitals and their application to biochemistry. A reminder that we've been talking a lot about different resonance systems, going all the way back to video 4 and up to video 19. And then after video 19, we talked about aromaticity. So in these videos now, we're going to be relying upon the knowledge background that we developed way back here in these earlier videos. Now, this we have written up here is an ester compound. And again, as we talked about back in video 6 or 7, you can have a bit of a resonance stabilization here. Here we have a pi bond between the carbon and the oxygen. This oxygen is sp2 hybridized, so it does have a lone pair of pi electrons. So as you saw us do a lot in the earlier videos, you can imagine this oxygen keeping both of the pi electrons in that bond for itself, giving it a negative charge, leaving carbon with an empty p orbital because oxygen stole the electron from it. But then this carbon has a lone electron pair. It can use that to form the double bond with carbon. But since oxygen put up both of the electrons to form this pi bond, it has a positive charge. So it's not a real strong resonance system, but there is some resonance stabilization that takes place there. Something else that is possible to happen, not very likely, but it can happen in small amounts, and that is for this carbon to lose a proton, so that the ester is very, very mildly acidic. And when this loses a proton, we have this structure. Incidentally, as we said before in the previous videos, this oxygen is sp2 hybridized, so it has a lone pair of pi electrons and sp2 electrons as well. It's the pi electrons, of course, that participate in any resonance that takes place. But now, here, the carbon loses a proton, so it keeps the electron from oxygen, giving it a negative charge. This carbon is sp3 hybridized. The one with the negative charge is sp2 hybridized. And that negatively charged carbon is called a carbanion. It has an orbital molecular diagram like nitrogen now. It has an extra electron. And what can happen is the electron from the s orbital can be promoted to a p orbital. Then it becomes sp2 hybridized. So the one s orbital combines with the two p orbitals, the ones that have the single electron, to form three equivalent sp2 molecular orbitals. The p orbital with the lone electron pair is not hybridized. So the carbon with the negative charge then, remember the sp2 molecular orbitals have 120 degrees between them, they're planar, and then the pi electrons are above and below that plane, and this is called a carbanion. Now it takes energy to promote, of course, from the s to the p orbital, but when that's done, that means carbon has a lone pair of pi electrons, so we can have some resonance stabilization of this carbanion. Again, the oxygen keeps both pi electrons, giving it a negative charge, leaving the carbon with an empty p orbital, but this now has a lone pair of pi electrons to make the double bond. This had a negative charge. It donates both pair of pi electrons, leaving it neutral. So here we have a resonance system where negative charge is here. Now it is here. It's dispersed, and the oxygen, and this oxygen here is left alone, so it has no positive charge. So you can form, the ester can be somewhat acidic to form this carbanion with the resulting stabilization of it. Now, instead of having oxygen, what happens if we have a thiol ester, if we put sulfur in here to have this compound? The sulfur will be sp2 hybridized, just like the oxygen, so have a pair of sp2 electrons, which we won't draw, but we will indicate the pi electrons. And again, the same kind of resonance situation. Like this, the oxygen keeps both pair of pi electrons, has a negative charge. This lone pair comes in to form the double bond, leaving the sulfur with a positive charge. But there's a problem here. And as we discussed in video number 25, here we would have a pi bond between carbon and sulfur. But now, for the carbon, its subshell is the 2p subshell. For the sulfur, it's the 3p subshell. So when we're trying to form this pi bond, you've got the mismatch of the pi orbitals. So this is very ineffective. So this does not happen as well in the thiol ester as it does in our normal oxygen-based ester. Now what about here with this carbon? Can this be acidic? And it certainly can. It's just the same thing. This oxygen keeps both the electrons, and remember our carbanion that we formed when this loses a proton has a lone pair of pi electrons. So we have this. Like this, same system as what we had right here. Same thing happening. This keeps both pi electrons. This donates its lone pair of pi electrons to form the double bond. This had a negative charge. It donated both electrons to form this bond. Now it has no charge. And in fact, with a thiol ester, you're much more likely to form the carbanion. And the reason for that is because of these mismatched orbitals here, this is less likely to occur. So the carbonyl group here, this carbon, is less likely to be involved with this, forming this pi orbital with the sulfur because of the mismatch of the pi orbitals. Therefore, it's much more likely to engage in this resonance, which means that with a thiol ester, it is easier to generate a carbanion than it is with the normal oxygen-based ester. And the reason is when you have an oxygen-based ester, you can have this kind of resonance, or you can have this kind of resonance. Here, with the thiol ester, this double bond here, because of the mismatch of the pi orbitals, is not likely to form. So almost all the resonance is here involving then this carbon losing a proton. So consequently, a thiol ester is better suited to generate a resonance stabilized carbanion. And that is, in fact, thiol esters can generate a carbanion far more easily under physiological conditions. And that's why, for example, for a molecule like acetylcoenzyme A, this is acetylcoenzyme A, you have the sulfur atom here. And that's the reason why. Because with that sulfur atom, then it's much easier to form carbanions under physiologic conditions. And steroids and fatty acids, they're all biochemically formed with enzymes that catalyze, as you see later on in your organic chemistry book, there's aldol-type condensation reactions, and there are also Claisen-type condensation reactions. These are all dependent upon an active carbanion. And in biochemistry, these types of reactions, the aldol-type reactions and the Claisen-type condensation reactions, those are how steroids are formed and how fatty acids are made biochemically. And the whole thing is predicated upon having coenzyme A with the sulfur atom here so that you can form the carbanion easily under physiological conditions. And again, the reason for that is when you get the thiol ester, this resonance here is much less likely to occur because this pi bond is much less likely to occur because as you've been speaking about in the past two videos, you have this mismatch of the pi orbitals. Carbon has a 2p pi orbital, sulfur has a 3p pi orbital. You don't get the overlap. You don't get the overlap occurring extensively enough to form a strong pi bond, as when you do when you have two 2p orbitals overlapping. For example, when nitrogen and oxygen form a double bond, or when carbons form a double bond. Here the pi bond can be of respectable strength. Here it is not because of that mismatch of the pi orbitals, meaning that for a thiol ester, the resonance that occurs will involve the carbanion. Therefore, with acetylcoenzyme A having that sulfur there, you can form those carbanions more readily under physiological conditions, and then that's what's behind then the biochemical mechanisms in aldol condensations, Claisen-type condensations, and those then are the organic mechanisms used in biological systems to form fatty acids and steroids. And again, that's why that sulfur atom occurs right there in acetylcoenzyme A, because of the mismatch here making it possible to form that essential carbanion. Okay, that's it for this video. Hopefully that was helpful to you. A lot of textbooks don't explain the significance of pi orbital mismatch and its applications to biochemistry. Here we did that in this video, and we also had a different application in the previous video. A reminder, the playlist for all the videos is at the website digital-university.com.